Okay, good. So Brian, thank you very much. Thanks also to Linda and to Mary Lou. It certainly is an honor to be here today and to be a part of the BEBRF for many years. You can't hear yet. Can't hear? I'll put it a little higher. How about that? Is that better? Better? Better. Everyone hear back there? Ah, good. Okay. I'll talk a little louder too. Okay. So, in any event, I was saying it's been an honor to be a part of the BEBRF for many years, and I thank Brian and Linda and Mary Lou for their great organization for this meeting. So, my topic, by the way, this is the National Institutes of Health. And my topic is the pathophysiology of blepharospasm, and I'm also going to talk about photophobia, which is a topic of recent interest and, of course, an important aspect of blepharospasm. So my question that I have to deal with is, what is the cause of blepharospasm? And when you have to consider that question, you really have to deal with what is the cause of any dystonia, and for that matter, what is the cause of any disease? And I think that the way that one has to think about that is by this particular formula. So this is P equals G plus M. Now the idea here is that P is the phenotype. And the phenotype means what a particular person looks like. And in this case, we're talking about blepharospasm. And then there's actually several G's and many M's that lead to a particular phenotype. G stands for genotype. Dr. Fahn talked about genes and the fact that that it is certainly part of really any disease. We are searching for them that are going to be relevant. He did comment that there is one gene known in relation to spread, that is the polymorphism in that TOR1A gene. And I just want to point out that some of the evidence for that comes from work that was supported by the BEBRF. A number of patients that were members of BEBRF participated in that study. So that shows the product of some of the BEBRF research. And then there are modifiers of the gene, so that it isn't only genes that cause disease, but there are important modifiers. And Dr. Fahn talked about some of them that are known, for example, the coffee, the sunlight. We know about gender effects, which are very interesting, and we don't understand at all why, in fact, women are more affected than men. Age is an important consideration, so there are a whole variety of factors that are relevant. We don't necessarily understand how they lead to the situation or how they influence the gene, but these are very critical. I think I'd like to mention trauma or some sort of infection. That also seems to be relevant. Very often at the onset of blepharospasm, there is some irritation of the eye, dry eye, blepharitis that has been demonstrated in many epidemiological studies. So that's also critical. And then plasticity. The brain is a very plastic organ of the body. That is, it changes all the time. And different factors can lead to those changes that go on in the brain. And I'll point that out to you in a little bit about its importance, particularly in relation to blepharospasm and other forms of dystonia. So Dr. Fahn talked about the epidemiology and genetics as far as what we know at the moment. I pointed out how they are highly relevant to causing disease. And let me just go on next to say a few words about anatomy and pathology. Presumably all of these factors lead to some changes somewhere in the brain that give rise to the particular disorder. And the question is, where is it in the brain? And you would think that we would know more about that now than we know. But let me tell you what we do know at the present time. First of all, there are neuropathological studies. We certainly need more of them to try to identify where one can find pathology. But at the moment, studies like this look for neuropathological abnormalities, and nothing has been found. So at the moment, even though we would like to be able to say, well, look, there are pathological changes in this particular spot, and that's important, really people have looked, and we don't find anything yet. Now, I think to a certain extent, we have to look more cleverly. There are probably, there are almost certainly changes that are relevant in certain places, but they haven't been identified yet. Now, another way of trying to find what the relevant anatomy is in secondary blepharospasm. That is blepharospasm that is due to a specific lesion in the brain. For example, a little stroke or something of that kind that is in the brain, and that can cause blepharospasm. And you say, well, maybe that's going to be similar to the primary or isolated blepharospasm that most of you have. And here is a study that was done by Buzz Jinnah and colleagues looking through the whole literature to see if they could find any similarity in the different cases in this regard. And basically they found 48 cases, but the lesions were all over the place. Almost in every part of the brain, they found different lesions that led to blepharospasm. So in the thalamus, in the brainstem, in the basal ganglia, in the cerebellum, in the cortex, all over. So to a certain extent, that means that there might not be a single place, or it might mean that there's actually a network of structures. The brain does work in networks. The brain is networked together. Things are all connected to each other in some ways, and it may be that any part of that network can give rise to the problem. We don't understand that yet, but this is the secondary blepharospasm information. Now, there's another way of getting information, and that's with neuroimaging. You'll hear more of that from Dr. Berman a little bit later, but I just point out the simple sort of structural types of imaging that have been done. One can do that with MRI. I don't know whether are you going to talk about some of the simple BBM? So I'll just point out that it'll be good if Dr. Berman can clarify this for us, because the point that I was going to make is that every study that's been done comes up with something different. And so I would hope that Dr. Berman will be able to clarify all this. In any event, here's one study that shows an increase in gray matter in the putamen. Here's another study that shows a decrease in gray matter in the putamen, as well as abnormalities somewhere else. And here's a study that we did at the National Institutes of Health that didn't find anything in the putamen, but found abnormalities elsewhere. So this has been very confusing, and I look forward to Dr. Berman to clarify all this for us. A little bit later. Now, what I'm now going to focus on is the pathophysiology. I think that's a little bit clearer. There are these fundamental abnormalities that we talked about in terms of the genetic background and other environmental and modifiers that are important, but somehow those together lead to pathophysiological derangements that lead to the eye closures, and that's what I'm going to talk about now with the pathophysiology. Now, there are a lot of studies that have been done in this regard in patients that have blepharospasm as well as other types of adult onset focal dystonias. And there are a number of themes which seem to be consistent through all of those studies. One of the more important ones is that there's a loss of inhibition. So in the nervous system, there is facilitation and inhibition. And there's a balance. That is, there's a promotion of activity in certain structures and an inhibition of function in certain structures. They have to be balanced. And what we see is that there's a loss of inhibition in many different areas, allowing for more activity to come out. And what that more activity would be is, for example, excessive eyelid closure. And so that's the sort of outcome that we think comes from a loss of inhibition. With respect to blepharospasm, this can be shown with blink reflex studies, and I'll show you that on the next slide. There's also some abnormalities of sensory organization. Those abnormalities give rise to very, very subtle abnormalities of sensation. They are so subtle that I'm sure none of you recognize it, but if it is studied very carefully, there will be some very mild abnormalities of sensation. And the underlying problem for that seems to be a loss of inhibition again. There is some inhibitory problem in the sensory processes as well as the motor processes giving rise to that. The third abnormality, which seems to be relatively constant, is an increased brain plasticity. Remember, I talked about that before as being a possible cause of disease. And in this situation, there is evidence for increased brain plasticity. And what that means is that there's a greater chance for the brain to change than there would be without this increased plasticity. So certain drivers, certain things that happen over and over again can potentially lead to a brain change. One of the ways of thinking about that is with dry eye or other abnormality in terms of the eye. People tend to blink more in that circumstance because dry eye is very irritating. And if you blink more over and over and over and over and over again in the setting of increased plasticity, that can drive a permanent change. So that's why plasticity can be potentially important interacting with environmental factors. Now let me show you the inhibition thing with the blink reflex. This is a study that might have been done in terms of a clinical test to help verify that you might have blepharospasm, because it's a very good test. If you give a shock to the nerve just above one eye, then the eyes will blink. This is called the blink reflex. And there are many ways of actually producing blink reflex, but that's one of the ways of doing it. And this is looking at EMG activity, the muscle activity induced by that shock. And let's look on this side first. In a patient who doesn't, in a person that doesn't have blepharospasm, you'll have a EMG response that has two components in the eye that has been stimulated and one component on the other side. This is the so-called R2, or the second response. And if you give a second shock at a short interval after that, you'll see that the second blink is smaller than the first blink. This is a type of inhibition, okay? The second blink is inhibited compared to the first blink. If you do this in a patient who has blepharospasm, this is blink 1 and this is blink 2. You can see that there's less inhibition. So this is some evidence for that decreased inhibition that I was talking about earlier. Looking at a number of patients who have blepharospasm, this is the less inhibition compared to a group of patients who don't have blepharospasm. So this is evidence for this loss of inhibition, this very important factor. So the current concept of the pathophysiology then is that there is hyperexcitability of the eyelid closure that comes from a combination of a permissive substrate like loss of inhibition that might arise from a genetic basis. That's the sort of reasoning that we think is going on at the moment. And an environmental factor like one that acts to increase the gain of eyelid closure, giving rise to a plastic change in the central nervous system control mechanism. Now, I'd like to briefly talk about photophobia. It is an important characteristic also of patients that have blepharospasm, and there is beginning to be some understanding of it and perhaps some sort of analogy that I'd like to mention in terms of the blepharospasm itself. So photophobia is where light causes pain or discomfort. That might be more properly called oculodynia than photophobia. Photophobia means fear of the light. And light can cause blinking or spasm of eyelid closure. The pathophysiology has been unknown, but I think there are some recent interesting ideas. So in terms of the way the eye receives light, there are a number of photoreceptors, the blue cones, green cones, red cones, and rods. They are the important photoreceptors that focus an image and give you things that you can see. But there are also other cells in the retina, and these are cells that contain an interesting substance called melanopsin. And these have other features. And some of the features that are important, they are involved in the pupillary light response, so that if you get little light in the eye, the pupil closes. They're also important for sleep onset modulation and entrainment of the circadian rhythm, that is the sleep-wake cycle. But interestingly, they are also connected into the thalamus and to parts of the brain that are relevant to sensation and pain. And this is now a possible way in which people can have photophobia, where this information from the melanopsin-containing cells can get into the trigeminal ganglion that has sensory function, can get into the thalamus where there are sensory receptors, and it can interact in the dura. The other group of patients that have trouble with photophobia, in addition to blepharospasm patients, are migraine patients, and they have abnormalities in the dura of the brain. So there are these interconnections here with sensory implications of what's going on with this type of input. The photosensitivity of different retinal pigments is indicated here in terms of the wavelength and the relative absorption. These are the red, green, and blue cones. This is the rods, and this is the melanopsin pigment. And this is where the center of blue is, over here. So you can see that melanopsin is really right in the center of blue. The so-called blue cone is actually a little bit out there, a little bit shorter wavelength than actual blue. So if there's, so the hypothesis now that ties into what I was talking about earlier is that there might be loss of inhibition in the trigeminal nucleus or the thalamic circuits for light stimuli. And if that's the case, there could be increased discomfort and heightened reflex blinking as a result of too much light input if there's loss of inhibition there as well as loss of inhibition in the motor circuitry. In any event, it is the case that decreasing light flux can help. So the question that is still somewhat controversial and is an interesting topic that perhaps we'll be talking about a little more later is clearly if you decrease the amount of light flux, that's better. Is it better to decrease a specific color of light that would be relevant to the melanopsin peak? And of course, the FL41 lenses are close to, but not exactly the same as the melanopsin peak. So it is conceivable that, that this type of lens would be useful for that purpose, but if it really is melanopsin, then it is a slightly different wavelength than the FL41, and that might be a modification that might be done sometime in the future. So the needs for the future is more research. Better understanding of the etiology may well lead to better therapies. Genetics may be an important help since that determines the substrate for each individual, and we clearly need to do more work in that area. Just in relation to more research, just to follow up Dr. Fahn's point, the BEBRF has been extremely helpful in terms of research over the years, and I think that that's been a very important function. I already pointed out to you how it has been helpful, but it's been helpful in many other ways. Dystonia Coalition is now a group of investigators interested in all the adult onset focal dystonias. And there's a new project, so-called Project 4, which will be starting relatively shortly, that will be focused specifically on blepharospasm. And we're hoping that all of you will help participate in that to move the field further forward. So thank you for your attention, and we'll look forward to the panel discussion.